I would like to start by thanking the organizers for the opportunity to present here today, and thank you to everyone listening. The data I am sharing with you are part of my recently submitted PhD thesis and are currently being prepared for publication. So this project was made possible due to collaboration between Jennifer Roger Lab, that has an extensive expertise working with the rodent-specific brain stimulation coils, and my primary supervisor, Alison Kenty, that has an extensive expertise in real-time in vivo brain imaging. At the time I joined Alison's lab, we were working on behavioral experiments combining the low-intensity intermittent theta burst stimulation with skilled reaching tasks, showing that delivering this type of stimulation prior to the onset of the learning task in mice, we can see increase in the accuracy of the performance, as you can see here in that red line. However, we were not that lucky in finding neurobiological correlates of these neuroplasticity-like effects in our biochemical assays. So going forward, we decided to look a bit more closely at some of the synaptic events using more powerful techniques of in vivo brain imaging. So when we think about neuroplasticity, synapse is a really good place to look at because synapses are very plastic. So these structures are points of communication between two neural cells, typically formed between a presynaptic axonal button and postsynaptic dendritic spine. And later in the conference, Dr. Alex Teng will be talking about some of the data from dendritic spines. But today I'll be talking to you about terminal axonal buttons, and I'll be talking about them in context of Alzheimer's disease. So as many of you know, Alzheimer's disease is a progressive neurodegenerative disorder that is characterized by extracellular deposits of beta amyloid plaques and intracellular neurofibrillary tangles. And some of the key hallmark pathology associated with Alzheimer's disease is synapse dysfunction that leads to synapse loss and ultimately cell death and brain atrophy. So in our lab, we use the APPPS1 transgenic mouse model of extracellular amyloid plaque deposition. And here you can see a cross-section through the brain of a 6-month-old animal. And these white dots here are extracellular amyloid deposits that are visualized with MOAB2 antibody. And as you can see, they are really quite pronounced in the hippocampal and cortical regions. And the data I will be showing you today all come from axonal projections that are found in these superficial cortical layers. So in order to gain an optical access to these axonal projections in vivo, we cross our APPPS1 animals with a transgenic line that expresses green fluorescent protein in a subset of their excitatory cortical neurons. We then perform a cranial window implantation whereby we replace part of the skull with the glass cover slip. And following recovery from this surgery, the animals are largely unaware of their new modification, but we do get an optical access to the underlying neural circuitry. We can then use a powerful 2-photon laser scanning microscopy to image deeper in the cortical layers at quite a high resolution. And here are just some examples of what we can see here when we look through the window with just a naked eye, and you can see the vasculature very clearly that we can use as a sort of a guidance system or a map that allows us to find the same region repeatedly. Here is an example of what you can see once you turn the microscope on. So you can see the green fluorescent protein expressed nicely and sparsely in some of the projections. Here we have a dendritic arc, but these thick projections, and the much thinner ones are the axons. When we then zoom in on the individual axons, we can see very clearly the individual processes. And again, looking a little bit more closer, we can visualize the individual axonal buttons at a really quite high resolution. And imaging these repeatedly will allow us to see that some of these axonal buttons are persistent over time, while others disappear or other ones are newly formed. And so we can get a range of readouts from these experiments. I will be talking today about density as the number of buttons per length, and a turnover as a measure of proportion of gains and losses between 2 consecutive imaging sessions. So we perform our craniotomy at 10 to 13 months of age. You can think of it as a sort of a midlife for these animals, and the pathology is really well pronounced at this stage. We allow 3 weeks for recovery after the surgery before we start imaging in 48 hours intervals, delivering the stimulation at the midpoint, and then keep on imaging again in 48 hours intervals. So looking at the density data first, so we have the wild type animals here on top and the APP-PS1 at the bottom. We can see that there are no significant differences in the density between the individual imaging sessions, which is good. So we can average the data per group and have a look at the differences between the wild type and APP animals. And we can see that at the baseline pre-stimulation imaging, there is no difference between the density of these axonal boutons. And when we look post-stimulation, the story is really much the same. So we did not see any changes in density in the wild types and neither in the APPs post-stimulation. So next we looked at the turnover of the axonal boutons. Again, here we have the wild type and APP pre-stimulation. And when we average these data, we can see that the APP-PS1 animals had significantly reduced the turnover pre-stimulation. And then when we introduced the stimulation, what we saw is that in both the wild type and the APP animals, there was a transient increase in the turnover for up to 6 days post-stimulation. And perhaps more interestingly so, when we graph the mean pre-stimulation data for the wild type animals here across the graph for the APP-PS1 animals, we can see that the initial lower pre-stimulation turnover was increased post-stimulation to the levels of the pre-stimulation of the wild type animals. So that was a very exciting finding. So overall, I showed you that the low intensity intermittent theta burst stimulation can induce neuroplastic response in our imaged excitatory axons. The axonal boutons that we imaged maintain their total presynaptic outputs post-stimulation. However, the targets of these outputs were highly dynamic. And the post-stimulation increase of the low baseline synaptic turnover in the APP-PS1 points to possible clinical application of the intermittent theta burst stimulation in Alzheimer's disease. And with that, I would like to thank my supervisors, our collaborators, and of course, our funding buddies. And thank you for listening.